The CSF is a sterile environment, therefore any organisms found are always indicative of a serious infection. A few of these infections will be explored today. The central nervous system consists of the brain and spinal cord, which work together to coordinate and control the mind and body. Infections of the CNS develop rapidly and are quickly life-threatening if not promptly diagnosed and treated. These infections most commonly include meningitis, encephalitis, and brain abscesses. Meningitis is classified by inflammation of the meninges or subarachnoid space and can be caused by a variety of pathogens, including bacteria, viruses, protozoans, yeast, and fungi. An array of viruses commonly cause meningitis, such as enterovirus and herpes simplex virus. However, the disease is most commonly caused by bacteria. Bacterial meningitis is one of the major causes of infection of the CNS and is usually spread via the bloodstream, but may also spread directly through the adjacent structures, shunt infections, and skull fractures. There are a few main species of bacteria that cause meningitis, and each tend to affect different age groups. E. coli, group B streptococcus, and Listeria monocytogenes commonly cause meningitis in neonates. Haemophilus influenzae commonly affects children under 5 years old, where Streptococcus pneumoniae and Neisseria meningitis affects people of all ages.
Methods and results. To diagnose a CNS infection, samples of the cerebrospinal fluid are taken from the lower back via a lumbar puncture. If the CSF appears cloudy to purulent, that indicates a definite problem, as normal CSF should be clear. This potentially indicates a bacterial infection, and a differential slide of CSF is observed. The presence of many polymorphs and/or mononuclear cells, generally greater than 80%, indicates an infection caused by pathogenic bacteria. The concentrations of protein and glucose are measured as additional confirmatory tests. A low glucose is observed due to its consumption by the bacteria, in addition to a normal to raised protein level. These tests ensure that the patient has a pyogenic bacterial infection and not an abscess, tuberculosis, or cryptococcal infection. Once the issue is determined to be a bacterial infection, the patient is empirically treated with penicillin and amoxicillin, depending on age, and awaits further results. The CSF sample is then cultured on 3 plates in 5% carbon dioxide, horseblood agar, chocolate agar, and McConkey. A swab from the site of infection is also used for a gram stain. The gram stain is the most important step for identifying the species. Since the CSF is sterile, any organism can cause a significant infection, but the major 6 organisms can be identified in a few steps. This is case number 1. This is the gram stain from case number 1, showing gram-positive rods. A gram-positive rod that grows equally well on horseblood agar and chocolate agar, but only minimally on McConkey, is likely a Listeria. This can be identified by observing its tumbling motility and beta homolysis on horseblood agar. A positive Aeschylean test and hipurate test would identify the genus, and an API Listeria would confirm its species. It's probably Listeria monocytogenes. Test it with ampicillin and penicillin G, but it is not recommended to be treated with vancomycin, gentamicin, ceftriaxone, or cefotaxime.
This is another case study. They came up with a gram-positive diplococci, so that's probably Streptococcus. If the culture showed alpha hemolysis on the HbA, set up an optician test. If it's optician sensitive, it's probably Streptococcus pneumoniae, but it's good to get confirmatory tests. In addition, it didn't grow on McConkey, which is enough evidence to support that it's Streptococcus pneumoniae. Antibiotic testing with ceftriaxone, cefotaxime, ampicillin, penicillin, and vancomycin should be performed and is usually resistant to gentamicin. Another Streptococcus was case 3. They showed up with a gram-positive cocci, so that again eliminates us down to Streptococcus. However, on the HbA it showed beta hemolysis, so that brings it down to Streptococcus agalactiae. To further identify the species, do another optician sensitivity testing. If it's resistant to optician, and if it tests for a B group in the Lansfield Group B test, it's probably Streptococcus agalactiae or Group B strep. Test on penicillin and vancomycin first, and from the penicillin results, you can infer ceftriaxone, cefotaxime, and ampicillin. This unfortunate case showed up with a gram-negative rod. This narrows it down to Haemophilus and Enterobacteriaceae. To identify the bacteria, do a catalase test and an oxidase test. If it's catalase positive and oxidase negative and grows equally well on MacConkey and HbA, it might be an Enterobacteriaceae. For our purposes, that just leads us down to E. coli, but it's good to do a confirmatory test. Conduct an API 20E. In our case, it came up with an E. coli. Then perform beta-lactamase tests for this organism and find sensitivities for cefotaxime, ampicillin, ceftriaxone, and gentamicin. It is always resistant to vancomycin and penicillin. This case study presented with a gram-negative rod, which is either Haemophilus or an Enterobacteriaceae. Again, do catalase and an oxidase test. If it's catalase positive and oxidase positive, the culture may be Haemophilus. And if it only grew on the chocolate agar plant, it probably is. Set up an XMV test or a satellite test for a broader result. If it only grew around the XMV disc, it's most likely Haemophilus influenzae. But check with an API NH. The culture can be shipped off for serotyping as well. It is not recommended to use penicillin, gentamicin, or vancomycin. Sensitivities to ampicillin, cefotaxime, and ceftriaxone should be tested.
Case Study 6. Case Study 6 presented with gram-negative diplococci, and there was no growth on MacConkey. The sample might be Neisseria or Moraxella. Further identification can be obtained by a sugar test where glucose and maltose presented positive and lactose and sucrose presented negative. If the sugar tests do not give a conclusive evidence, an API NH can be performed and they can give us a positive ID. And for case 6, the API result was Neisseria meningitis, the second most common cause of bacterial meningitis. These tests can be performed separately or in an API NH, not recommended to treat with gentamicin or vancomycin, but it can be sensitive to cefotaxime, ampicillin, penicillin, or cefotaxime. The report. The laboratory report clearly shows how much more detail, and the cell counts biochemistry identification tests show how Neisseria meningitis was identified from the patient's lumbar puncture. According to the results in the laboratory and doctor's report, it is clear that the patient is suffering from an infection of Neisseria meningitis in the CSF. Although Neisseria meningitis was isolated, any bacteria found in the CSF is immediately indicative of an infection as it is a sterile site. Meningitis is a bleak disease to experience, but with prompt notification and reliable identification methods, a patient might be able to make it to tomorrow.